This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 842, recorded on December 14th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here we have a nice, bright, sunny sky, and the temperature is 45 degrees Fahrenheit. And in Celsius, that's, your rain Norwegian weather app, 8 degrees Celsius. So pretty nice. Weird.
Let's see what we have. Let's see how comparable. It sounds weird. It's a sunny out, 11 degrees Celsius. Very nice day. Also joining us from Madison, New Jersey, Brianne Barker.
Hello, it's great to be here. And it is a very nice day. Of course, same weather here that Vincent has, but it's lovely out there. Doesn't seem like December.
Yes, it's strange weather. That hasn't gotten really cold yet, right?
Right.
Maybe at night goes down to 4 degrees. All right. This is your midweek TWiV fix. And yesterday, I was on a call with Theodora Hatsuanu. We're discussing already the next edition of Principles of Virology. Can you believe it?
Ooh.
Because you have to hand the publisher the final manuscript a year and a half before they plan to publish it. Isn't that crazy?
Yeah.
That's amazing.
So we said they want us to hand in the next version in 2023. And that's too soon. I'm sorry. I said we need to at least sort out some COVID things and get answers. So we devoted to delay. Anyway, while we were on the call, Theodora sent us their latest preprint, and we will talk about that today. It's pretty interesting. And, I try to stay away from preprints, but it's hard. They're seductive, right? And this one is relatively straightforward. And the assays are essentially neutralization assays with pseudotyped viruses. So, as they say, what could go wrong? Right? So this is plasma neutralization properties of the SARS-CoV-2 Omicron variant. We have three co-first authors, Fabian Schmidt, Frauke Muksch, and Jiske Weisblum. And this is from the Rockefeller University, the PI labs, Michel Nussenzweig, Theodora Hatsuanu, and Paul Benash. And also on this paper is Christian Gebler, who we had on Immune, right, Brianne?
Yes. And he was talking about some early data that sort of relates to this. This paper includes a cohort of patients that has been involved in a few papers from this group. All of those papers have been really interesting.
So we actually talked about similar results last Friday from South Africa, from Pfizer. This it takes it a little bit further. And so if you recall from Theodora and Paul being on TWiV, last time they were on, they talked about PMS20, which was it's a VSV pseudotype with the spike of SARS-CoV-2 into which they had added 20 amino acid changes that they had identified that led to resistance to neutralization with monoclonals and convalescent serum.
I'm going to be rich here and remind you that VSV is vesicular stomatitis virus. It's another virus which is very amenable to putting things into its surface. And in this case, that's been done with this spike protein. And that is then called a pseudovirus because it's not really VSV, it's not really SARS-CoV-2, it's a pseudovirus.
And they say, here's the sentence, 20 RBD, receptor binding domain, and N-terminal domain mutations into the spike protein. And I once said to Theodora is on the textbook, and she knows that a mutation is in DNA. And so I think she has little participation in the writing, probably, and doesn't have time to even go through it that finely. So that is very similar to Omicron. which has many changes in the spike. So they use those two viruses in the neutralization as well as ancestral virus. And as Brienne said, they have a nice collection of sera from patients, convalescent patients, patients immunized, patients who recovered and were immunized. And so that's what they're looking at. And they're doing neutralization assays. So I guess this paper was submitted on Friday, I think. And so that was the 1 week mark of Omicron about? So it's just,
I think maybe it was maybe a couple weeks.
2 weeks. It was the Thursday after Thanksgiving.
Okay.
Wasn't it on Thanksgiving?
That's what Thursday of Thanksgiving.
Yeah, Thursday of Thanksgiving, I think the WHO report started happening.
Okay. All right, it's basically you pull out these sera from the freezer and you do dilutions and you mix them with the PMS20 and all the other viruses, the pseudotypes, and you do neutralization assays. So here's what they found.
Just after the 2 week mark then.
It's good for 2 weeks to get all the data and then write a paper, right?
That it's more than good. I'm really amazed.
Yeah. It takes a long time to do experiments. And often you do an experiment for a year and then you take it months to write and you have multiple authors and you go back and forth. So
it might be okay that she missed the one thing about the mutation. If they were able to make all of these viruses and do all of the experiments and write it up in 2 weeks.
Well, the last time she sent me a preprint, it was the same thing. I said, Theodora, why do you say this? She said, do we? So she didn't even know it. That's fine. All right, so PMS20 and Omicron, pseudotyped Omicron, right? The spike of Omicron pseudotyped into viruses. They had to make that, but I guess it doesn't take very long. Is substantially resistant to neutralization by plasma compared to the Wuhan ancestral virus. And here are some numbers. So we have convalescent people. They recover from infection. The plasma is collected about a month after infection. So they have 50% neutralization titers. And they, for example, for Wuhan, this sera would be 2,616. That's a nice titer. Then for PMS20, it's 49, so much reduced neutralization. And for Omicron, 68. So the higher the number, the higher the titer, neutralization titer of the serum. And they also present that as a fold reduction, 60-fold reduction for PMS20 and 58-fold for Omicron compared to Wuhan, HU1. Okay. After 6 months, now 6 month sera from the same patients, the neutralization titer drops. So we have 1,678 for HU1, 28 for Omicron, and 42 for, PMS20 it doesn't matter. 28 and 42 are basically the same as the 1 month. It's just the ancestral titer that's gone down. So those are reductions of 37-fold for PMS and 32-fold for Omicron. And then they also did 1 year after infection, and then they look similar. Right.
Yeah. So the idea is in people who were infected, they are making antibodies that are less able to neutralize at particularly 6 and 12 months. After infection of both this lab-made spike and the Omicron spike, which has some similarities to the lab-made spike.
And, as we discussed on Friday, we really don't know what that means in terms of protection against disease, contrary to some people on Twitter who are taking these percent reductions and using them to calculate vaccine efficacy. Can't do it. We don't know what these mean. And, after the paper we did on Friday with on immune correlates of protection for respiratory syncytial virus in non-human primates, some of the correlates are not neutralizing antibodies. They're FC-based functions of antibodies. So I think you have to be – people get very upset about these results, but I think you have to be careful. And then, of course, there's T cells too, which – anyway. So those are people who didn't get vaccinated. And next they look at sera from – plasmas from vaccinated individuals. All right, so the NT50, 7,600 for HU1, 60 for PMS20, and 95 for Omicron. This is 1.3 months after that second vaccine. I mean, that's a compressed, right, 3-4 weeks between the doses, which we know is not a good thing. 187-fold reduction for PMS20 and 127-fold for Omicron. 5 months later, 43 for PMS20 and 126 for Omicron, which is 58-fold and 27-fold reduction. So vaccination, the antibodies induced by vaccination don't do a great job against PMS20, which they already knew, and Omicron spike. That was an mRNA vaccine, right? Yeah.
That's with the mRNA vaccine, yes.
Yeah. So then they also looked at J&J vaccine, the adenovirus vectored vaccine, which they make the editorial comment. These plasmas were particularly poor at neutralizing variants. 1 month after vaccination, that's a single dose, right, of the J&J. Anti-50 values of 588 against the ancestral, which is way lower than the mRNA titers, less than 25 for both PMS20 and Omicron.
If you look at the graphs here, this graph is striking.
It is.
I find the numbers a little bit tricky to keep straight in my head on this, but the graphs are very obvious.
But that titer of 588 still conferred protection against disease in the original clinical trial against the ancestral virus, right?
Yes.
All right. And they say, there was a lot of variation from sample to sample. Some of the individual plasmas didn't have any neutralizing activity whatsoever. All right, so now we look at the combination of being infected and then vaccinated, which we know from their work and others improves the breadth of the antibody and also the neutralizing titers, right? So they have convalescent individuals who have gotten a dose of mRNA vaccine, or vaccinated individuals who got a third dose, right, over 6 months after the initial series. So previously infected and vaccinated, we now have 388,000 50% neutralizing titer against HU1, 15,000 against PMS20, and 8,000 against Omicron. So 8,000 is as good as, let's see, the recovered people against the ancestral strain. It's amazing, right?
Yes, it's as good as either the recovered or the vaccinated people against that ancestral strain.
Yeah. And over time, this is pretty stable. So 5 months, the titers were stable. So that's an interesting observation. What else here? So there's an increase there as opposed to a decrease. And now we have people who got a third mRNA dose, Pfizer, a month before they took the serum, the NT50 against HU1 was 65,000, against PMS20 1,500, and against Omicron 3,800. So these are still better than when they had two only. Much better, right?
Yes, it certainly speaks very well for a third dose here.
Yeah.
These data really make a third dose look quite good.
26-fold increase for HU1, 35-fold for PMS20, 38-fold for Omicron. They say we conclude that boosting with mRNA vaccines dramatically increases neutralizing titer and breadth for individuals who had previously been infected or vaccinated. So, and this is kind of the conclusion from the studies we talked about on Friday, but here, of course, the addition of there's different sera sets, more extensive, I think 169 different sera, and a comparison with PMS20. And I don't know if you remember, when they made that virus, they said, well, this probably would never emerge in nature. And something similar to it has, which is, so this still remains really surprising to me that we have continuous emergence of antigenic variants. I guess, the unprecedented number of people infected and also the sequencing we're doing is all playing into that, right?
I think so. We have so many people infected. It's hard to think about how much bigger the population is than it has been in many big past pandemics. And so just the number of infected people is way more than we've ever had. And if all of those people have a lot of virus, that's more than what we're seeing in a lab situation.
So I asked Theodora, I said, okay, now you get let's say a third dose. Your titers have now gone up. You now can neutralize PMS 20 and Omicron. What happens in 6 to 8 months when the titers go down? And she said, well, there are two possibilities. One is if you get infected, then your memory should capture this increased breath, right? That's correct. So now the increased breath given by that third dose should be captured in your memory B cells, right?
Yes, exactly.
Okay. But then I said to her, do you think we'll need another boost in 6 to 8 months? And she said, well, if you want to really stop transmission, because we have so many unvaccinated people around, there's a lot of virus, that's what you'd have to do, is keep boosting every 6 to 8 months, because we know that when the titers are very high, you don't really get infected at all. So that would be an interesting scenario.
It would. I guess the question is, are we trying to protect against infection or are we trying to protect against disease?
Well, of course, we would like to protect against disease. And what this does against disease remains to be seen, right? So obviously we're going to find out in a number of months how, a third dose does against disease when the titers wane, when they contract. But I don't think you always want to prevent infection, right? But maybe now that you have a situation where there's so many unvaccinated people, maybe you'd really like, I don't know. What do you think?
Well, I think the one other piece of this that I notice is when I look at their patients who were infected and then vaccinated, those individuals have seen the antigen 3 times. And the data that they're showing here is 6 months after that third encounter. And they're showing that the antibody response is pretty stable in those people who have seen the antigen 3 times. It's not just improved, it's also improved and sort of stably at this higher level. And so what we can't tell here is how long the boost response will stay at this increased level. If all of these third encounters give you something somewhat similar, then maybe you would expect the same persistence, the same 6-month persistence that we see in figure 6A or 4A to be what we see for 4B if we pulled figure 4B out longer. We just don't have the time to know that yet. I think that would also have quite a bit of bearing on this in what we're protecting against.
Is it possible that 2 doses give you a certain level for 6 months and it contracts, but 3 doses last longer?
That is what Shane Crotty seemed to imply, where he was implying that there was a qualitative difference as well as a quantitative difference to 3 encounters with an antigen.
I see.
And so that was what he mentioned when he was last on TWiV, and I have been actually trying to go down the rabbit hole of finding data on that ever since.
But go ahead. Yeah.
Well, it's a little bit off on a tangent, but I've been having a question in my mind about statements where, this says increasing the neutralizing titer and breadth. And do we always know that it's breadth or could it just be depth? I mean, is the evidence about breadth because of variance or is it depth?
That's what Amy said on Friday. How do you know if you just have a high titer, it's just going to be good at neutralizing, and it's not really breadth? Yeah, that's a good point.
Right. I think their other paper talks a little bit more about breadth, where they're looking at the ability of the same sera to neutralize more variants. I think it's harder to make that claim here, where they're not testing against as many different viruses. But if I recall, in one of their other papers, they actually were able to see hybrid immunity where they had infected and then vaccinated individuals get responses that were so broad that they could neutralize the original SARS-1. And so I think that shows breadth. I'm not quite sure if we see breadth here.
Yeah. Also, there was a paper we did last week where they looked at the actual affinity of the antibodies for the RBD by affinity measurements, and they could see with time that the affinity increased, and so there you could quantitate it, right?
Right,
in terms of antibody diversity, right?
Yeah, but that doesn't necessarily tell you breadth. If you got a much stronger response against one variant, that wouldn't tell you that you're necessarily getting a stronger response against a different one.
That's true. So the breadth you get from trying different spikes from different coronas.
Exactly.
Right? Yeah.
And so when the antibodies are undergoing affinity maturation, you get a broader set of antibodies that come out of that.
So if you're measuring the affinity of the antibodies, that could be an indication that it's becoming broader, right?
So if you're measuring the affinity of the antibodies, that means affinity maturation has happened.
Right.
And when affinity maturation happens, you get some that have better affinity against any individual variant, but you also get versions that will work against many different variants.
I guess, Kathy, they call increased breadth, PMS20 and Omicron. That's their definition. But we would like to see more, right? Because they may not all be equal, right? But I suspect we will see that in other studies. I'm really interested to see as these levels contract. I don't know when they will. Maybe it will take 12 months. Then what's the protection against hospitalization and death? Is it better than before? I don't know, because you can imagine in those first days after you're exposed, your memories, B cells are making antibodies. If they're really better at neutralizing the virus than antibodies against the ancestral, then maybe you would have an improvement in protecting against disease.
I would also like to know the same thing, but I really hope that it is very hard to answer that question because 12 more months later, I hope that the amount of virus is a bit less.
Yeah, the number of infections hopefully will be much lower, so it'll be harder to do that, right? Yeah. Yes. I hope so. I do hope so. Okay, there you go. That is, wait a minute. No, cancel. That is our preprint for today. And now we have a paper, which actually Rich Condit sent to me last week. And he said we should do this, but make sure Brianne is there. And I said, well, she'll be here Tuesday. And he said, well, I won't be there, but you don't need me.
Oh, I think this paper was a make sure Kathy's there kind of paper.
Yes, Kathy, of course. But he wanted your immunological comments. But he's not here, but that's fine. So I think we have enough people to do it. This is Science Advances. chadox1 interacts with CAR and PF4 with implications for thrombosis with thrombocytopenia syndrome. Yeah, I thought Kathy would like this, right?
Right.
Yeah.
Had you seen it before?
No, I hadn't seen it yet.
Cool. So we have two co-first authors, Alexander Baker and Ryan Boyd, and three co-second authors, Daipayan Sarkar, Alicia Tejera Crespo, and Chun-Kit Chan. The last author is Mitesh Borad. And in there, among the authors in the middle is Linda Coughlin.
Colin.
Sorry?
Colin. Linda Colin.
When I said Coughlin to her, she said, that's fine. I know.
Okay. And I wrote to her and I
before.
said, am I pronouncing it wrong if it's Colin? And she said no.
Colin?
Yeah, something like that. You don't
Colin.
say all those vowels or those consonants in the middle.
Well, I also did a guttural thing, which... but okay, Linda
Something
Collin,
like
who's been on TWiV before, a long time ago, and was at Oxford working with the people there on this vector for other purposes, I think influenza immunization. This comes from large number of groups: Mayo Clinics, Arizona State University, Cardiff University, Michigan State University, University of Illinois, Glasgow Center for Virus Research, Emory. No, not Emory. Sorry. What else? AstraZeneca, of course. University of Maryland. That is where Linda Collin is.
Collin. I looked up the email. She says, here's how to pronounce my surname. It is like Colin, like the sound a crow makes, or Colin, Caulin.
Okay.
Not cough like respiratory, though I often think this is quite cool and to have cough in the name of a PI who works on respiratory viruses. And it's an Irish name and spelling. And in Irish, it means daughter of the cloaked one.
Daughter of the cloaked one.
Colin.
Okay, Colin. Linda Collin. Like Cullen, right?
Almost, yeah.
Brian Cullen.
But it's more like Colin.
Colin.
Yeah.
Okay. Colin. Anyway, this is a very lovely paper. And Rich was not just interested in this TTS and its association with immunization with COVID ad-based vectors, but he thought the experimental approaches were pretty cool. So we'll see if we can do this justice. All right, so thrombosis with thrombocytopenia syndrome, TTS, has been seen in some recipients of the AstraZeneca vaccine following the first but not the second dose. Also in the Janssen vaccine, which is an adenovirus 26, species D, adenovirus type 26. And does anyone know offhand roughly the frequency that we're talking about? I don't remember. I didn't look it up.
I thought there was something in here that gave a number, but no, I don't remember.
So this is similar. It's a similar syndrome to heparin-induced thrombocytopenia. So what is? I had to look it up because I don't usually think about these things. So this all involves PF4, which is platelet factor 4, which is a chemokine that is released from platelets when they aggregate, and it promotes blood clotting by moderating the effects of heparin-like molecules, which would antagonize clotting, right? PF4 binds to heparin, and that complex is the antigen in heparin-induced thrombocytopenia, which is an autoimmune reaction to when you give people heparin as an anticoagulant. In some cases, you can have autoantibodies made, and that's what HIT is, thrombocytopenia.
Yes.
So
I had a question for Kathy.
Yeah.
Are these types of issues, either TTS or HIT-like issues, known to be observed in other types of adenovirus, either infections or in adeno studies otherwise?
So I really don't know anything much about, nor do a lot of people, about natural adenovirus pathogenesis in humans, with respect to that aspect of your question. I do know that there's a couple of references in here. One that it said, the very recent finding, and that's actually a review article a reference to two things, references 9 and 10. But there's an earlier paper that's one of the same first authors, Othman et al., adenovirus-induced thrombocytopenia, the role of von Willebrand factor and P-selectin in mediating accelerated platelet clearance. And that's from 2006. So Paul Young pointed that out last spring on Twitter when people were wondering about adenoviruses and thrombocytopenia, and he pointed that out. So it's something that has been at least observed, but I didn't have time to go back and look at those papers to tell you more about it.
Yeah, I was actually really struck on that front by a sentence in the abstract that says, as part of the largest vaccination campaign in history, ultra rare side effects not seen in phase 3 trials have been observed. And I hadn't really thought about it, about being the largest vaccine campaign in history. But I realized that probably is correct. And how that would allow us to see ultra rare side effects that might not have been seen in many of these other situations before. And so I thought that was really interesting and was something that really made me think.
So you also, in these TTS cases induced by the ad vectors, we also see antibodies to PF4. And so that's the reason people say it's similar to HIT, because in both cases you get antibodies to PF4, and that, of course, is part of the problem. So they're looking at this, in this paper, they solve the structure of the chadox vector and try and figure out where PF4 is binding. And they also do some other things that are kind of tangentially related, but that's, to me, the most interesting thing. And they raised the possibility later that you could modify the vector so they don't bind PF4 anymore, right? I think that would be a cool outcome.
So just a reminder, the chadox vector is the chimpanzee adenovirus Oxford. That's where the ch-ad-ox comes from. And that is the basis of the AstraZeneca vaccine, which then now has the name AZD1222 or Vaxzevria. So most people may have thought of it as the AstraZeneca vaccine. We early on encountered it as chadox 1, and so it has these multiple names.
So they say in the introduction, which is interesting. So, human Ad5 had been previously developed as a vector, and they found that would bind platelets or coagulation factor X, and they say this causes the vector to be degraded and has a reduced therapeutic index. So this made the field develop alternate vectors with less seroprevalence to get around that. And that's where, in part, chadox and the human ad D26 comes out. And that's kind of interesting, right? That was the driving force there. In part, I think there are other driving forces as well.
And that will probably come back at the end because they talk about how this is important for vector degradation. And you could sort of imagine as we get to the mechanism that will come up at the end of this paper that you can see how the vector degradation is happening.
So they first solved the structure of chadox1 at 3 angstroms by single particle cryo-electron microscopy. And I love they write, has the quintessential icosahedral adenovirus capsid structure. It's lovely. They have a beautiful picture of it here, the complete capsid and
the complete capsid lacking the fibers.
They don't have the fibers, They couldn't resolve it, and they had to do that separately by crystallography.
Yes, and they use a word that I had not yet encountered, which was peripentonal.
Oh, yeah. The around around the pentons.
Kathy, do you want to comment on the quintessential icosahedral capsid structure?
Well, 12 5-fold icosahedral vertices is where the penton base structure is found. It's got ordinarily has the fiber knob. It's a lovely structure.
And so they see the pentons and then they see the hexons, which are the trimers of a single protein that make up the bulk of the capsid, right? And those are arranged in groups of 5 and 6, correct?
Right.
And a part that is important, it's going to come back later, there are parts of the hexon called the hypervariable regions. And I suppose, Kathy, those are known to be more diverged than the rest of the protein, correct?
Correct. Yeah. And they're seen as loops. I think, we see them in figure 2. They're the loops that differ when they try and match the structure of the chadox 1 to ad5 or chadox 1 to ad35.
And of course, also in this structure, they see not just the pentons and hexons, they also see other proteins that are in between, like P9 and P8, right?
And 3, and
Yeah. All right, so then they solved the structure of the fiber by crystallography. You can see the knob at the end. So it's a long extended alpha helix. It's a trimer. And at the end, there's a knob which is involved in binding to the cell receptor. And they model, so this is done with chadox. So they use that structure to model the interaction of the fibers with CAR. So CAR is the fiber attachment protein for some adenoviruses. For human adenoviruses, would that be fair to say?
Some human adenoviruses. The Coxsackie adenovirus receptor protein is its name, but of course it doesn't exist in the cell to be a receptor for Coxsackie virus and for adenovirus. But yes.
Yeah, that's why poliovirus receptor was renamed after we pointed it out to CD155. Yeah, so sad. So they made models of various adenoviruses, including C5 is a CAR interacting adenovirus. B35 is a CD46 interacting adenovirus. It does not bind to CAR. And they can look at the contacts that are made. And I think essentially they find that what they think from the modeling is that the chadox fiber binds CAR, but not CD46 by looking at the models and the interactions and so forth.
Right.
I like that they included B35, the one that does not bind CAR, for comparison here. I found that really helpful as someone who does not spend a lot of time looking at these.
Right. So they already had the structure of the Ad37 fiber to CAR, and then they modeled the chadox1 or the Ad5 binding to fiber based on that. And so based on that, they conclude that chadox1, the model is consistent with it binding CAR, and then some other studies. But yeah,
they do some actual binding studies with the proteins, and they do surface plasmon resonance, which is a nice way to do affinity measurements among ligands and receptors. And they say this proves that the C5 and the chadox fiber binds CAR, but not CD46.
Right. Or also desmoglein, which is another receptor for some adenoviruses, human ads.
DMG, right. So there are 3 different or more than 3 for human ads?
Oh, well, there's at least those 3, and then there's other things that may be factors or, may not. It's complicated.
Like the integrins that are entry receptors, okay. And then they validated these in cells by they take cells that produce CAR and but none of the other known adenovirus receptors, and they can add virus to them and measure the affinity, inhibit it with antibody against CAR, but not CD46. So CAR is the receptor for chadox, not 46 or DSG2. Okay. So then they move on to PF4, where they say it's been reported recently that chadox may interact with PF4, and that's part of this clinical presentation of TTS. So they...
just a reminder, PF4 is platelet factor 4.
Right.
And this is a small molecule that is secreted from cells to be involved in blood clotting. And so the fact that it is secreted and is generally extracellular, again, will be important later.
So they again use surface plasmon resonance, a way of measuring binding, to see if PF4 in fact interacts with very pure preparations of adenovirus 26, Ad5, and chadox1. And they also have
chloride purified.
Cesium chloride purified, yes. Highly purified. Also vaccine preparations of chadox1 and AZD, 1222, which I presume they got from the company. So you want to compare to what you make in the lab to what is actually being injected in people because they're not the same, right?
Right.
So chadox1, Ad5, and 26 all bind to PF4 with nanomolar affinities of 660, 789, and 301. So the lower the number, the better, right? And AZD1222, the vaccine, has a similar affinity to PF4 as the virus that they made in the laboratory. They confirmed that this is specific binding by using an antibody against PF4 in these surface plasma and resonance experiments. You put the one component on a chip, and then you flow the other component over it in microfluidics, and then you shine a light on it, and when it's a laser, I believe, and when the proteins interact, the way the laser is reflecting changes, and so you can measure that as affinity. It's just amazing, right? All this in a tiny thing you can't even...
So it's basically almost like how much the chip deforms
Yeah.
from that binding, and you can sort of see the binding on and off by looking at how the light beam moves because the chip was deformed.
And so then you can add an antibody in, and it should inhibit the interaction.
And and it changes the shape of the curve when that happens.
Yeah.
Exactly.
So what they find is that when bound to PF4, antibodies bound to PF4, antibodies can still bind PF4 when it's complex with chadox, which is part of the whole idea of what's happening, right? PF4 is binding the virus, and the antibody is still able to bind it.
Yeah, so the antibody and the virus must bind to PF4 in different spots.
Right. And so they did this binding experiment with different salt concentrations, and they found with increasing salt, they get less PF4 binding, from which they conclude, and this is an electrostatic interaction, meaning it's involving plus and minus charges. So then they can look at the surface of the particle that they've calculated and calculate its electrostatic potential, right? What areas have a lot of plus, what areas have a lot of minus. And then so they can say there are areas where PF4 could certainly bind, right, by this charge analysis. And so then they do simulations of PF4 binding, knowing the charge of both. Can we simulate where this thing would bind? And what they find is that PF4 frequently contacts the capsid between the hexons, and most commonly at the interfaces of 3 hexon where there's a lot of negative charge. So the PF4 is positively charged. There's a cluster of negative charges there, and that is allowing PF4 to interact.
Yeah, we're sort of just seeing that there's this very unlucky charge matchup here, allowing for this binding.
And they look at one particular binding event, which they call an exemplar binding event. Shows the electropositive faces of PF4 on its longest axis oriented to face the negative electronegative potential of the surrounding hexons. Stuff happens, right?
Mm-hmm.
It's amazing. And so then they say what amino acids in the vector are important for this, and they can identify those that are part of this electronegative region, and they can identify contact residues in the hypervariable regions of chadox1. That's why I said, remember, hypervariable region, because that's where they think this thing might be interacting.
And that's so important because now you could potentially start changing those amino acids.
Yeah.
And looking at whether that changes the ability of this to be immunogenic, bind to CAR. They can do all of that in terms of the CAR binding with modeling.
Yeah.
But then they might be able to remove this ability to bind to PF4.
Yeah. I was wondering, do you think they'll do all of that? I mean, I don't know how frequent this TTS is, right?
I think that they will do all of that.
Oh, yeah.
I can imagine, if we think about all of the different places where adenovirus vectors are being developed, the knowledge that this is a problem could be a pretty significant detractor from that development. And so making a version that you can show does not have this potential, to me, sounds like something that is surely going to happen.
Yeah, just having seen the field over the years, all the modifications they've made for gene therapy vectors and now for vaccines.
Okay, that's good. So they're responsive. That's good. And also this is a nice example to people who are vaccine hesitant. You say, okay, look, we monitored safety, we learned, and then we fixed it.
Exactly.
What more could you ask for, right? All right, the last experiments involve heparin, which is part of the HIT disease in people, right? It binds PF4,
right? But not part of the TTS,
right? Not part of TTS. So they wanted to know what effect heparin binding to PF4 would have on the ability of PF4 to bind chadox. And they have an anticoagulant drug called fondaparinux, probably saying it wrong, which is made up of a heparin pentasaccharide that binds PF4. So it's an anticoagulant by virtue of binding PF4. And so the presence of fondaparinux reduced the number of binding initiation events of PF4 with the capsid surface. So the implication is that fond binding to PF4 can inhibit the binding of PF4 to the capsid.
And they sort of say, well, since this is a heparin pentasaccharide, that saying that heparin could inhibit this binding.
And of course, they did the heparin experiment. Heparin pre-incubation of PF4 with heparin also inhibits binding to chadox as well. So they think in these cases, the negative potential of PF4 is being reduced by either fondaparinux or maybe heparin. And so
when you say negative potential, you don't mean in the electrical sense. You mean the negative consequences, right?
Yes, of course, yes.
Okay.
No, not electronegative.
Sometimes I can be too literal, so.
Yeah, I understand. So this weakened electronegative potential may reduce the ability of PF4 to form an electrostatic association with chadox. So does that mean you could give people fondaparinux? Or if it's by the time they get the TTS, it's too late? Probably it's too late, right?
Well, they kind of talk about that. The current clinical guidance advises against the use of heparin for the treatment of TTS. And although the data suggests that heparin might inhibit the proposed interaction between chadox and PF4, it doesn't provide any insights as to the effect on heparin in patients after they develop symptoms or so forth. So, they say it's important to continue to adhere to current clinical guidance pending further studies on the role of heparin in TTS.
So they say that, all right, so now a little discussion. Platelets, the surface of platelets has CAR. So they say, don't be tempted to link CAR and chadox binding to TTS. Because adenovirus platelet aggregates are trafficked to the liver where they're sequestered and degraded, as I think Kathy mentioned before. So probably that doesn't have anything to do with TTS.
Yes, and in fact, I also really liked the sentence after that because they point out a previous study that could induce thrombocytopenia in mice by giving a really large dose of adenovirus. Mm-hmm. They then point out that requires a dose that is 7,500 times higher than that given in the vaccine. And that, in that case, given intravenously instead of this vaccine, which is given intramuscularly. And so I thought that was a nice point to make that comparison.
Yeah. But even in that case, there were no thrombotic events. There was just thrombocytopenia. And then another study in rhesus macaques showed the opposite effects, longer clotting times, probably as a result of diminished platelet count or depletion of circulating factors. Therefore, it's unlikely that direct association between chadox and platelets drives thrombotic events. So they're trying to minimize that as a cause of TTS. But they say we've shown that PF4 and chadox interact, and this suggests the mechanism for TTS. PF4 binds with high affinity to chadox that they grew in the lab, and also the chadox vaccine, and that may drive it. And it is not specific to chadox because PF4 also interacts with AD5 and AD26 with similar affinity. Interesting.
And also the fact that they had that highly purified virus interacting with PF4 argues against there being anything else in the vaccine formulation coming from cells or something like that was causing this.
And so it makes it important to think about doing similar modeling and looking for those similar patches on AD26 and AD5 if you want to use those in order to remove this binding as well.
Current evidence indicates that TTS presents similarly to HIT, appears to be driven by anti-PF4 autoantibodies of sufficient affinity to cluster PF4 and create a multivalent, presumably higher avidity interaction between the antibody Fc domains and Fc gamma receptors on the platelet surface. So I want to know, Brianne, why we are making autoantibodies against PF4.
So I found they have a supplemental figure 10. And I found supplemental figure 10, which was their model here, to be incredibly helpful on this because I read this and I said, and then I looked at the figure and thought about it and I said, oh, of course, now I get it. And so I think that what they're trying to say is that in either case, there are some of these antibodies and they are, we're getting PF4 clustered in some way. So we're making clusters of PF4. It could either be clusters of PF4 with the virus in the case of TTS, or clusters of PF4 and heparin in the case of HIT. And so we're making some big clusters of PF4. And in getting rid of those clusters, the degradation part that came up earlier, those clusters are getting phagocytosed. This molecule that is extracellular, PF4, is now getting into cells. And when it's inside of cells, it's getting presented on MHC class 2. And that's helping you to turn on some T cells. Normally, T cells cannot respond to PF4 because PF4 is not inside of cells. But when we make these big clumps, we internalize the PF4, we turn on T cells. And now that the T cells are turned on, the T cells can actually feed back on B cells and say, yes, make lots of antibodies against this. And so we have to have that T cell intermediate turning on a B cell to start making the autoantibodies, which we didn't have before. And so the key here is that the virus or the heparin is clustering PF4 and allowing it to get phagocytosed.
Cool.
And again, supplemental figure 10.
Yeah, I didn't get that far, and I'm just now looking at it. It's great.
It's really great, and it's really helpful in helping you to understand this.
So why do we have B cells against PF4 to begin with?
We have B cells against basically everything.
So they're not eliminated like self T cells.
We have slightly different ways of eliminating self B cells and self T cells. And it's a little easier for B cells to fall through the cracks. B cells are only tested against things that are found in the bone marrow. So perhaps PF4 is not in the bone marrow. So the B cells can't test against it. We can't eliminate them. And the way that we normally protect against those B cells getting activated is that the matching T cell never gets activated. We're okay. But in this case, we're activating the matching T cell. this probably only happens rarely in individuals who are predisposed. One thing that they'd have to have is the right MHC
Right.
in order to present just the right peptide to make this whole thing work.
Got it. They also point out that these symptoms manifest 5 to 24 days after vaccination, which corresponds to the timeline for a secondary antibody response.
Right, exactly. And so that gets to that whole idea that it's not just you have the PF4 binding to the virus and that's the whole thing. You actually need that internalization, those T cells and those B cells actually getting help in order to have the autoantibodies formed.
And the other point is they say TTS is much less frequently observed following the second dose, suggesting that, similar to HIT, anti-PF4 IgG is not long-lasting, and any plausible mechanism should be prominent in the first but the second dose.
That one I found a little harder mechanistically to really understand. And the main thing that I could come up with is that the people in whom this is happening are genetically predisposed and are going to largely have it happen to them the first time.
Yeah, they mentioned that this needs to be looked at in vivo. If you can make adeno-PF4 complexes and give it to an animal, will they get thrombosis? That would be good.
Right.
That would be useful. By the way, they're postulating that when you get vaccinated, small quantities of the vector enter the blood through minor capillary injuries caused by the injection, which I could totally understand, right?
Yeah, absolutely.
Not intravenous injection by accident, but a little bit of vector getting in broken capillaries. It makes sense. And that's where the vector PF4 complex forms. And they say, this is their hypothesis, and now they say this explains why it's so rare, because it requires a series of low-frequency stochastic interactions. First, between adenoparticles entering the blood, which is not going to be a lot, and then monocytes and or B cells, which may only occur in individuals who are predisposed to the generation of anti-PF4 antibodies. So there's a subset of people that are predisposed to do that. Not everyone makes anti-PF4, right?
Mm-hmm, exactly.
That's the idea. Okay. And then they talk about modifying the vector to try and eliminate electronegative residues in the hypervariable regions. But of course, as Brienne said, you have to make sure that you don't compromise the antigenicity or anything else as well. But that could be fun.
Right. And they point out that it could sort of be killing two birds with one stone, because you could potentially reduce the TTS and reduce the anti-vector immunity, which is the problem that they have if you have pre-existing immunity kind of thing, I think, is what they're getting at.
Yeah, absolutely. If you stop this vector from getting phagocytosed, if you stop internalization, you are going to stop making antibodies against PF4, helping out those B cells, but you're also going to stop some of the anti-vector immune response.
An alternative they suggest is maybe out there's another adenovirus with lower PF4 binding that we could use.
A naturally occurring one, yeah.
Yeah, that would be interesting.
There's at least 50, 60 odd adenoviruses.
Yeah.
So.
All right, so go ahead.
I realized that I missed one opportunity when you were talking about the quintessential icosahedral structure. For our holiday door decoration contest, my lab made a mouse adenovirus that's really cool. And I'll send you a picture of it, Vincent.
Nice.
Maybe you can use it for the show image, but I'm quite proud of it. They built it on an icosahedral base, and
nice.
Yeah, it's pretty neat.
That sounds very cool. I think this is a great show of the importance of some of these basic science things coming back to help us understand what we're seeing in clinical things. I like what you said about being responsive to the vaccine issues that we've seen.
Yeah, this is a basic science paper, and it's all happened since the chadox was licensed, right? I presume, in response to those TTS events. So that's really good. Right. Why don't we go to some picks because I would like to finish at 3:30 because Brianne and I have an immune this afternoon.
Okay.
So let us go to picks of the week. And Brianne, what do you have for us?
So I have something from Time. I really liked the cover that I saw. And when I went looking for it and found this link, there was, in fact, a little article that accompanies that cover. And the article is also great. And so Time yesterday picked their 2021 Heroes of the Year. This is in addition to some other picks they made, but really I'm interested in the heroes of the year, which were vaccine scientists. And so there is a version of the Time cover that has Caitlin Carrico, Drew Weissman, Barney Graham, and Kizzmekia Corbett. It's a lovely picture of all of them. And the accompanying article kind of goes through some of the backstory of them putting together the SARS-CoV-2 vaccine and some of their backstories. And so I found it to be a good, perhaps if someone didn't want to read all of A Shot to Save the World, they wanted a somewhat shorter version of an article. This goes into some of the same types of things and is also just a really nice way to honor the hard work of these vaccine scientists.
Cool. I
Nice.
hadn't seen this yet.
Very nice.
Thanks.
Yeah. I saw the cover, but I hadn't seen the article. And I was even more excited when I saw the article.
Kathy, what do you have for us?
I picked something that was in the Michigan news thing today. It's about snake dietary diversity. There's a nice article about it and a short 1-minute video. And the gist of it is that when there was the mass extinction that took out the dinosaurs, that made an opportunity for snakes to really expand their dietary things. And I'm trying to find out, I saved some of the key remarks. It wiped out most plants and animals, but this is a doctoral student's work showing that snakes responded to the mass extinction with a burst of dietary diversity. And so they did this study using both genetic evidence with dietary information from field observations to, here's the key part, preserved museum specimens, and made mathematical models and looked at how prehistoric snakes lived. And so there's one quote: "A lot of the stories of evolutionary success that make it into the textbooks, such as Darwin's famous finches, are nowhere near as impressive as some groups of snakes." And that's Daniel Rybowski, whose doctoral work this was. So I just thought it was an interesting take on what happened in the mass extinction with respect to snakes, who probably don't get enough attention.
Yeah, the mammals get all the attention, right?
Yep.
So they just, their diet expanded because they had more opportunities. There's not competition as much, right? That's the
right.
idea.
And it talks about how they, some of them started, worms and other snakes and just all kinds of breadth possibilities. So, yeah.
There are snakes that eat other snakes.
Yeah. Yeah.
It's a pretty one here. The American pipe snake is a predator of elongated vertebrates such as snakes, legless lizards, and legless amphibians. Cool stuff. Very nice. This is from University of Michigan.
Yeah, I think it was from the Record. It's an online thing that I get every day. And somehow there was a picture of a snake with a little link to the video, and then I was hooked.
So there are people at Michigan who study snakes, right?
Mm-hmm.
This doctoral student was a Michigan student.
Right. Yes. Yeah.
Cool. Very nice. My pick is it's actually an Instagram. I've been following this group on Instagram for a while. It is the Alfred Wegener Institute. And this is and I also provide a link to the webpage. This is an institute in Helmholtz, and they send out expeditions to study, climate change, environmental changes, and so forth. They have a bunch of ships, and they have stations, at the poles. They have observatories, and the Instagram feed is cool because they'll put out a picture of scientists working on these ships or out on the ice or on the snow or whatever it is down there and working in their stations and all this equipment. And it's pretty cool that, we're on our way to this location. It's going to take us X days to get there, and we're going to do this. So I think it's pretty cool. The logistics and the infrastructures involved. These are huge ships, right? They have pictures of the ships, and you can click on them and see what they do. They're full of equipment. There's one ship, the Youthorn, is on 140 days a year on a lake. They study nutrients and salinity, turbidity, and so forth. So they have a bunch of these ships. They have various stations, and it's just fun to check out their pictures that they put up, which are not at all like pictures that we would. I think that's the thing that got me the most, is that there's a lot of different ways to do science, and this is really an extreme because you have huge ships and stations. You have to go to different parts of the world, and it's a big deal to get there, and then you do your experiments. So it's a lot of fun. So we have the website and the Instagram. And I just got one this morning. I forgot what the picture is, but it struck me, and I had been meaning to pick this for a while. So if anything, you could do the Instagram thingy. I think that's pretty cool.
Definitely different than I usually look at on my Instagram. I want to.
Yeah, the thing about this is that if you follow it, then they give you suggestions of similar things, and you have, you can go down rabbit holes of following all kinds of similar stuff. It's fun. We have a listener pick from Niklas. Hello from Svalbard. If you don't know where Svalbard is, it's way up north. To put into virology context, it's one of the places where they have tried to search for the Spanish flu. Christy Duncan wrote a book about when they were allowed to search for it in the permafrost at a cemetery here, hunting the 1918 flu. Haven't read it, but as I understood, they didn't find anything in the samples. I would like to share a pick, a children's TV series that I saw when I was a kid, and I loved it, still do. The series is a French cartoon dubbed to many languages, including English. There might have been some mistakes in the translations, though. Notice chickenpox was referred to as a bacterial infection, but it was correct in the original French. It's about the human body, and I think it was a great way to introduce kids to biology and what's going on in our bodies. Or for adults that like cartoons, it's called Once Upon a Time ... Life. What I think is the coolest is that in all its small details, it's very accurate and goes into complex areas with an easy-to-understand narrative. You get a description of the lymphatic system, and the lymphocytes are little flying spaceships. That's just perfect. So much so, I still connected it and drew help from it in high school, and I've heard of med students that think the same even in their higher studies. Even now, when I started with Brianne's YouTube lectures, there were things that popped up and that were explained, of course, not in the same depth in the show. I don't know if any of the TWiV panel have seen it. If so, what did you think of it? I thought maybe many of the listeners would be interested to show their kids or watch with them, and he provides a link on YouTube. Where it for now has all the episodes in English, that might change if copyright watchers shut it down. Anyway, I think you can order it on Amazon as well. Thanks for all the shows and talks. I'm currently also trying to go through Brianne's and Vincent's lectures to understand a bit more. Also wondering if there's a good way to understand papers more. I find some of the presentations of statistics, results, and graphs troubling. For example, concepts of confidence intervals. Do you think it's just a matter of reading more and going to Wikipedia when I'm scratching my head? Passionate people that are willing to help and share are to be admired. So thanks, all of you. Again, have a nice day, Nicholas. I haven't heard of this before. Have any of you?
I have not, and now I want to watch it.
Yes. Yeah.
So what suggestions would we have to understand papers better?
Well, it's a good idea to at least try to go to Wikipedia when there's something that you don't understand. For example, today I realized that my understanding of surface plasmon resonance is really sketchy. And I just didn't have enough time to go into depth. And I used Rich's trick of going to Wikipedia, and it was kind of way over my head. So I resolved to find some better explanation about that. But how I'm going to find that, I'm not exactly sure. But so that is one thing. If you want, send questions to the authors if you think it's something that they really could have explained better, and then maybe they'll realize what they needed to have done better. There were a couple things in the first paper that we did that I, again, being too literal, had a hard time with. So I don't have much else to suggest, when you get stuck on stuff, other than to try and look at it.
Yeah, I think that, practice is always something that helps here. The only other piece that I could add is that you mentioned that you find the presentation of statistics results in graphs, particularly as your trouble points. And I think that with some of my students, when they are finding results troubling, it's because they look at the results and they aren't sure how the results that they see in the graph correspond to the words they're seeing in the text. And sometimes that's a little bit of, that's a place where you should actually ask questions. And that's a place where that is ripe for discussion. Sometimes I will look at a graph and say that my interpretation of the graph does not match the author's interpretation. And I often tell my students, don't look at that as a place where, oh, I'm just a student, I don't know enough. Look at that as you've identified a point of contention and a place where you could have a discussion with the author. And so I think sometimes people come to that point and say, oh, it's because I just don't know what I'm doing, when in fact it's because you know exactly what you're doing and you're on exactly the right track.
Yeah, that's exactly what happened to me with these surface plasmon resonance data points. They would make some conclusion, and then they would reference a figure that was just one of those SPR graphs, and I would look at it and say, I don't know where they got that number, so yeah.
Yeah, I think looking at other sources helps. It could be a website like Wikipedia, which has gotten much, much better over the years. Today I looked up, PF4 for a quick way of finding out, instead of going to the literature, which would be great also, but then may have its own difficulties. Wikipedia is referenced, so you can check that out. But also, if statistics are an issue, you go watch a video or two on statistics. There are plenty of good courses out there. Don't be afraid to take some time to learn what you need to do. If a paper really means a lot to you, learn, because you'll reuse statistics for sure. So today we have a lot of options you don't have to go to the library and get a book out and read, right? Which we did in the old days. You can find it online. And
yeah.
So
I guess what I'm trying to say is it happens to all of us.
Yeah, for sure.
We're reading a paper and we hit a wall of something that we don't really know, and have to spend time to figure it out.
For sure.
Especially with confidence intervals, as you mentioned.
Yes.
That is a challenging concept.
No, we have to look things up. I mean, and I often go to the primary, depending on how much time I have, I go to the primary literature, but that can take a while. You can get stuck in the rabbit holes of references and references, right? Which can be very frustrating. And, but if you have time, that's a great way to do it. But I always think of people who say they listen to TWiV, and when they encounter something they don't understand, they stop and they Google it to try and get more information. And that's a great way to say, what does this term mean? What is HIT? These guys didn't define it. So I think all of those things work.
Yeah.
All right, that is TWiV 842. You can find the show notes at microbe.tv/TWiV. Send us your questions, comments, pics, TWiV at microbe.tv for the months of November, which is over, December, which is almost over, and January. All donations made to Parasites Without Borders will be matched and returned to Microbe TV. So parasiteswithoutborders.com, that's Daniel Griffin's nonprofit. If you scroll down a bit, there's a little button that says donate and gives you all the information. And of course, Microbe TV has recently become a nonprofit as well, 501c3. So if you'd like, you can donate to us. You can go to microbe.tv/contribute. I have to fix the donation page. Daniel showed me some clever tricks that he uses that I need to implement. But meanwhile, you can go there. And if you give $1,000 or more, you will get from me a signed copy of Principles of Virology, 5th edition, signed just by me, because I can't be running around the East Coast to get everyone's signature. But that's cool. A number of you have taken me up on that. That's very nice of you. We appreciate your support in helping to build Microbe TV as a communication company by scientists. Kathy Spindler is at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.